Thanks a lot, Sophie. Thanks a lot, Amalia, for the invitation. And of course, Catherine, who suggested my name to present today. It's a great pleasure to join this fantastic group. And I'm looking forward to meeting you all in the real life as well. So as Sophie said, the topic of my talk will be dealing with PPARα as a sexually dimorphic drug target in NAFLD. Like I said, as most of you know, non-alcoholic fatty liver diseases are major public health issues, and it's a range of diseases that starts with fat accumulation in hepatocytes and can progress to more severe stages and irreversible diseases. So, of course, there's a lot of association with obesity and diabetes. And there's also a lot of research dealing with these different stages of disease, because different factors that are leading to the irreversible stages of the disease, such as fibrosis, cirrhosis, and cancer, are not very well understood. And of course, there's no approved treatment for NASH, which is the dangerous stage of the disease. And one more thing that has to be said: there are not very many reliable biomarkers for NASH. So most of the time, patients have to be biopsied, which is not easy and very costly. So we started this project when talking to Pierre, whose lab is in Toulouse, and he's an endocrinologist. And he has a great expertise in sexual dimorphism and great knowledge in male predominant metabolic traits as well as female predominant metabolic traits, which is not my expertise, but I would like to refer to a recent publication from this group by Blandine Tramin and colleagues in Diabetologia, where you would find very many details regarding these specific traits. I'm a little bit more aware of work that's been done on mice. And this is one reference, data from one reference from Aldo and Lucie's lab. It's a huge effort they've done, because they've screened the metabolic status of very many different mouse strains. And as you can see, that in dark, you've got the reference for male mice, and in orange for female mice. And there are very, very significant metabolic differences between male and female, which depends on the type of strains. More specifically, there are also many different excellent publication on liver sexual dimorphism and possible link to diseases such as NAFLD, of course, from Adriana Magghi's group, and Adriana will be speaking afterwards. So I'm sure she will mention some of the work that's in this publication on the link between amino acid homeostasis and estrogen receptor signaling. There are also a number of evidences that liver sexual dimorphism is also very sensitive to both circadian biology and microbiota. This is the work from Frédéric Gachon's lab in cell metabolism as well. This work from Aurel Besse-Patin in gastroenterology also identified PGC1 alpha as a key actor in sexual dimorphism, especially in NAFLD. That's from the group of Jennifer Estal in Montreal. And there's also this publication in Nature Medicine, sorry, I forgot to mention, regarding the effect of thermoneutrality on NAFLD in mouse models. So what we did together with Sarah, and as Sophie said, our manuscript is now published in Gut, is that we investigated the effect of very different diets on metabolism and NAFLD in mouse. So what Sarah did is that she compared 5 different diets for their ability to induce obesity, glucose intolerance, and steatosis in vivo. And what she could see, and you can see it here, is that different diets such as high-fat diet, choline-deficient high-fat diet, and Western diet or Western diet plus glucose and fructose all induce sexually dimorphic responses with a much stronger body weight gain in males than in females, and many other differences that I will talk about today. So the first point that we also assessed is that the different diets induced change in glucose homeostasis, and that's also more striking in males in blue than in females in red. As you can see it on the high-fat diet response, or here the choline-deficient response, and also Western diet response. Then we looked at the liver, and we scored the steatosis, inflammation, and also other aspects of liver biochemistry. And what you can see on this slide is that we've got very, very significant differences between male and female mice exposed to these different diets, with always a much stronger steatotic phenotype in males than in females, and also a stronger inflammatory phenotype in males than in females, especially in the Western diet. Then we used gene expression profiling to try to estimate the contribution of the diet and of sex to the liver gene expression profile. And as you can see from this PCA, which is an analysis that ranks the different impact of the diets and sex on the global gene expression profile in the liver. The major difference that occurs is related to sex. So the sex is a major driver of the gene expression profile in the liver. And you can also guess that there are effects of the different diets on liver gene expression profile. But this is less, it's not as strong as it is for the impact of the difference between females and males. And so what we can say is that there's a very robust female and male biased hepatic gene profile that's independent from the diet. So it's represented here for 3 diets: a control diet, a high-fat diet, and a Western diet. And as you can see, there are significant differences between male and female that are specific of the diets, but there's a core of differences that are specific for female and for male, independently of the diet. And we try to figure out what are the pathways that are strongly impacted. And amongst them, of course, you can identify what was quite expected: steroid hormone biosynthesis, and basically steroid hormone homeostasis, but also other aspects of liver's biology, including coagulation cascade regulation, platelet activation, bile acid homeostasis, but also functions related to fatty acid homeostasis, such as linoleic acid metabolism, peroxisome activity, and so on. We performed some lipidomic analysis, which also pointed out at lipid homeostasis as strongly impacted by the diet and by sex, and the interaction between sex and the diet. And we also performed genome scale modeling in collaboration with Adil Mardino-Blu's group, which really pointed at lipid homeostasis as possibly dysregulated in a sex-specific way during the progression of NAFLD in response to high-fat diet or Western diets. And as examples of such genes that are sexually dimorphic and related to lipid homeostasis, you can see one of them very, very strongly different between females and males. That's elongase 3. And we know from experiments performed with whole body knockout that elongase 3 is involved in obesity and NAFLD development. but also other genes such as FSP27, or also called SITE-C, or ACOT3, and also PNPLA3, which are dimorphic. And people in the field know that PNPLA3 is a very important gene in the field of NAFLD. So we then thought perhaps some of our favorite genes, which are PPARs, transcription factors that connect together lipid homeostasis and the regulation of gene expression, might be involved. And as probably most of you know, there are three PPAR isotypes, alpha, beta, and gamma. They're not expressed in the same tissue, and alpha is highly expressed in hepatocytes and in most tissues that burn fatty acids. It's very important for fatty acid catabolism, and all of the different PPARs are drug targets. And because PPAR alpha is highly expressed in hepatocyte, and because there was some preliminary evidence and also other works reporting sexually dimorphic response to PPAR alpha activation, we thought it could be a good candidate for what we observed in unbiased approach. So what is PPAR alpha? It's a ligand-induced transcription factor from the nuclear hormone receptor family. And it's a transcription factor that's very active during fasting. So a while ago, together with Walter Wally, Alexandra Montagnier, when she was in the lab, made a liver-specific, hepatocyte-specific deletion of PPAR alpha, and provided strong evidence that PPAR alpha was very influential for the response to fasting and many other aspects of PPAR alpha biology. But if you look at the fasting response, which is a massive transcriptional response in the liver, with 8,500 genes are significantly impacted by fasting. 1/3 of them depend on hepatocyte PPAR alpha. And we looked at the functions that were regulated. And as you can see from this graph, of course, the peroxisome-related genes are impacted, as PPAR encodes for peroxisome proliferator activated receptor. It's one very, very important aspect of PPAR response. But also many other aspects of lipid homeostasis are regulated by PPAR alpha in hepatocytes, including mitochondrial fatty acid oxidation. So another way to look at this very important PPAR-dependent response is this unbiased analysis that we've done together with Catherine Postic and her lab, when we were looking for molecules that were specifically induced by glucose as opposed to fasting, and where we showed actually that FGF21 was one of the single mRNAs that responded to both stimuli. But what happens in response to fasting, as you can see here on the heat map, is a very, very strong response. And if you look at the gene ontology specific for fasting, PPAR alpha and PPAR signaling pathway comes out first. And from different experiments, we know that PPAR alpha is really the core of this, responsible for the core of this big response. And what happens during fasting is that free fatty acids from the adipose tissue become a very important substrate to make ketone bodies. And this response, this ketone bodies biosynthesis, depends on hepatocyte activity and on PPAR alpha dependent transcriptional response. Together with ketone bodies, PPAR alpha also is very key to the transcriptional control of FGF21, which is an hepatokine, an hormone that's coming from hepatocytes and plays a very important role in the adaptation to fasting. and controls many aspects of systemic response to fasting. So these are two important marks of PPARα dependent response in hepatocyte: the ability to burn fat to make ketone bodies, and the ability to produce FGF21 from the hepatocytes. We also showed that these mice lacking PPARα in hepatocytes are prone to fat accumulation, and that's enhanced by high-fat diet feeding, but it's already quite clear upon controlled diet feeding. And PPARα not only plays a role in the control of fatty acid homeostasis, but it's also an important regulator of the inflammatory response in hepatocyte, as evidenced by Bart and his lab for many aspects of this. So what we've done with our gene expression profile from the different diet experiments is that we perform network analysis, which are shown here below, for most of the nuclear receptors, basically all of the nuclear receptors that are expressed in hepatocytes. And we identified using these networks that are here that the top 50 genes around the plot here, that are related to PPARα expression in male mice are not as connected to PPARα expression in female, which marks for a sexual dimorphism in this network, that we could not observe such a massive difference for PPAR beta and gamma, which are shown here. And this was done in collaboration with a very talented bioinformatician called Una Bloom, who is working in CNRS in Rennes. So this data were pointing at PPARα as a possible regulator of some of the sexual dimorphism that we've got in liver during steatosis. So to challenge a little bit this concept, we looked at the responses to Western diet feeding in both wild type and knockout mice lacking PPARα only in hepatocytes. And we've done this in both males and female mice. So as you can see, you've got different responses between male and female. In male, actually the hepatocyte specific knockout did not gain weight such as the wild type mice, while in females we had the opposite, and we had knockout mice were more prone to gain weight, while the hepatocyte specific knockout were less prone to weight gain. Moreover, we looked at gene expression as well, and as you can see here from FGF21 expression, we have a very significant impact of Western diet feeding on FGF21, and that's very different between males and female. And if you look at the protein level, it's also very different. This response depends on hepatocyte PPARα, but is different between sexes. When we looked at steatosis, upon controlled diet feeding, we had a significant difference between males here and female here. The male are prone to spontaneous steatosis, while females are protected. And upon Western diet feeding, both sexes showed steatosis. To further investigate this, we performed fasting experiments, both in males and female mice. And we observed some common responses to both in males and females, but also some very significant differences between males and females, with a more spectacular impact of fasting on the response, the PPARα dependent response in females. Then we looked at the response to a pharmacological agonist, and that was done after we discussed with Bart, who told me that pemafibrate was a very, very specific agonist to be used in vivo to address this point. And he was really right. As you can see here, when we treated male knockout mice and female knockout mice with the PIMA fibrate, we observed no change in gene expression that could reach significant level in males, and only one gene was induced by PIMA fibrate in knockout mice in female mice. So these are hepatocyte-specific knockout, which means that all of what happens in response to PIMA fibrate in vivo in the liver depends on hepatocyte PPARα and not on other cells in the liver. And as you can see now from the response from wild-type mice, we have a massive response to PIMA fibrate treatment in male mice, as well as in females, but many differences between males and females, with some specific response in males and some responses that were only statistically significant in females. So we thought, regarding mouse work, we had quite significant and convincing data regarding the sexual dimorphism. And then we thanks to a collaboration with Hervé Guilloum and Christophe Bureau from Toulouse Hospital, we could, together with Pierre, we could do this analysis on a human cohort of men and women with different ranges of NAFLDs. So again, this is done on correlation analysis and correlation networks, and we've got a very dense and tight network for PPARα related genes in men that we could not find in women. And we also done that for FGF21. Again, we've got a very dense network for genes correlated with FGF21 in men that we could not see in women. So this suggests that the PPARα related network is quite significantly dimorphic in men and women, as well as we've seen in mouse models. So while it was not new that there are very many differences between men and women during NAFLD, and again, some of the work regarding that was done by Philippe Lefebvre in Barth Lab in Lille. What was new here is that we had a network related to PPARα and women that were dimorphic. So that's where we are for PPARα. Dimorphic as a sexually dimorphic target in mouse and human NAFLD. And what we are now doing is we're trying to investigate the molecular basis for such dimorphism, and possibly the cell autonomous crosstalks with other nuclear receptors and other hormone sensitive pathways. But we think this is probably very important for pharmacological responses, but we're also interested in non-autonomous regulation of hepatocyte PPARα activity and its role in sexual dimorphism. And I'm going to try to show you some very recent data to let you know a little bit more about that. So this is a work that we've been starting together with a consortium that we built together with Dominique Langin in Toulouse, Catherine Postic in Paris, and Zubir Amri from Nice, to study the dialogue between organs, and especially the dialogue between adipose tissues and liver. And through the progression of this work, I developed a collaboration with Achim Lass and his group. He's based in Grasse, and he provided us with mice lacking a rate-limiting enzyme that's expressed in white adipose tissue called HEGL, and a very specific white adipose tissue knockout that allowed us to investigate how much during fasting, as I told you, fasting is very important to induce PPAR alpha dependent response, such as ketone body synthesis and plasma FGF21. And thanks to the collaboration with Hashim and his group, we could assess how much of the response to ATGL deletion and impaired lipolysis from white adipose tissue would impair PPAR alpha dependent response in the hepatocytes. And the responses we've got were very, very clear. So that's what happens in mice that are either wild type or lacking ATGL only in white adipose cells. During fasting, wild type mice release free fatty acid and glycerol as a consequence of triglyceride hydrolysis in wild type mice, but not in the knockout mice because they lack the rate limiting lipase for this. And as a consequence, and as expected, there's no substrate or less substrate to make ketone bodies from the liver. So this could be only a metabolic response, but there's also less induction of FGF21 during fasting. And this was already known by different groups. But what we showed here is that beyond FGF21, there's also a massive transcriptional response in the liver. And this is a bit like what happens in PPAR alpha dependent mice. Upon feeding, you can hardly see a difference between wild type and knockout mice, but fasting reveals a massive difference between wild type and adipose tissue knockout mice. So we thought, okay, it's a good start. Let's go and see what happens next. We performed unbiased gene expression profiling, and we identified many, many different things. And I will just focus on one cluster, cluster 5 here, which is highly induced in wild type mice and not in knockout mice. This is typical of PPAR alpha response. But let me remind you, it's not a PPAR alpha knockout, it's an adipose tissue ATGL specific knockout. And this very specific cluster is enriched in genes, 300 genes, involved in fatty acid catabolism and enriched in a response element for PPAR alpha. And these are typical responses of these genes that are known to be PPAR alpha target genes, such as FGF21. As you can see, if you fast the ATGL mice, okay, all of these targets for PPAR alpha are induced in an ATGL dependent response. Then we compared the response we've got in ATGL adipose tissue knockout to PPAR alpha hepatocyte specific knockout. And we identified a very strong correlation when comparing these two very different models. And this correlation points at PPAR alpha dependent, a very, very well-known PPAR alpha dependent response, including lipid metabolism processing and also response, inflammatory response on regulation of cell death. So this was suggesting that free fatty acids from the adipose tissues were released to the bloodstream and activating PPAR alpha in hepatocytes. And to challenge this concept, we used a molecule that's very well known by people in the field of white adipose tissue and brown adipose tissue biology. It's a Cl compound that's a very specific ligand for beta-3 adrenergic receptors that are expressed in mice and control white adipose tissue lipolysis, as well as brown adipose tissue browning. And we use this molecule to challenge adipose tissues and to test the contribution of, again, the contribution of ATGL to a response that we could possibly see in the hepatocyte. So to start with the ATGL adipose tissue knockout, as you can see here, the Cl treatment elevates free fatty acids. That's okay. That's the response of white adipose tissue that's impaired in ATGL knockout. It also induces ketone bodies and FGF21 very significantly and in a TGL dependent way. And that's what happens in the hepatocyte. We have an HEGL-dependent response of typical PPARα target genes. And at this stage, what's very important to understand is that these experiments have been done in the fed state. So to us, it suggests that adipose tissue lipolysis that is not that is inhibited by feeding, but activated very acutely by the CL treatment is sufficient to induce a response in the hepatocyte. And that's suggesting that PPARα is sensing primarily the secretion of free fatty acids from the adipose tissues. So we've done the CL experiment again, but this time in mice lacking PPARα in hepatocytes. And this time, we had a very good induction of free fatty acid release by CL in both wild type and knockout mice. But the ketone body production and FGF21 production was impaired when PPARα is absent in hepatocytes. And of course, all of the transcriptional response is altered in the mice lacking PPARα in hepatocytes. So we really think this data, together with an extra transcriptome analysis that also confirms a very large set of genes are induced by CL in a PPARα dependent manner in vivo, strongly suggests that pharmacological activation of lipolysis by adipose tissue activates PPARα in hepatocytes. And quite unexpectedly, what we've also seen is that we have a phenotype in mice lacking PPARα in hepatocytes. We have a phenotype that can also be seen in the brown adipose tissue. I'll be very quick, but I will show you this. This is what happens when you treat wild-type mice with the CL compound, browning of the brown adipose tissues. That's impaired in mice lacking PPARα in hepatocytes. It's also true for some of the genes that are induced that are induced during browning, such as UCP1 and elongase 3, and as you can see in protein level as well for UCP1. And this fits together with some data showing that mice lacking PPARα in hepatocytes are unable to tolerate cold exposure as compared to their wild-type littermates. So we think that PPARα is not only very important in hepatocyte, but it's also very important to coordinate inter-organ crosstalks to get in this setting with white adipose tissues and brown adipose tissue. So one of the key questions that still remains to be addressed is whether all of this crosstalk that we have now evidence between PPARα in hepatocytes and brown adipose tissue activation relies on FGF21 secretion from hepatocytes, because several groups have provided evidence that FGF21 is an important hepatokine in the context of browning. So here we are, second conclusion. ATGL-dependent white adipose tissue lipolysis is a key signal for hepatocyte PPARα activity, and some of our ongoing work relates to this, the understanding of whether such signal and such crosstalk with white and brown adipose tissue might be sex-specific and contribute to sex-specific NAFLD, and also whether FGF21 is underlying these responses. And this work has been done by Anne Fougera, who was a postdoc in the lab, and from now 3 days, she is a permanent scientist in the lab, and we're really very pleased for her that she got such permanent position with us. So with this, I would like to thank all the people who've done the work. Most of the experimental work on sexual dimorphism have been done by Sarah, and the work on browning and adipose to liver axis has been done by Anne. But Arnaud, Marion have done a lot of work on PPARα, and the project with PPARα hepatocyte-specific knockout was started by Alexandra when she was in the lab, before she joined my friend Pierre and his group. And I'm also very thankful to all of these collaborators, especially Pierre and Dominique in Toulouse, Catherine in Paris, and all of the other collaborators, including Thierry, Bart, Sophie, and Hashim, who's helped us with different aspects of the work, Adil with the bioinformatics, and Maeva and Christophe, who helped us with the human work. And of course, I want to acknowledge the contribution of the different facilities at INRAE and at INSERM. We're really lucky with the facilities in Toulouse. And I thank you for your attention.